The vitamin C tablet in your medicine cabinet is ascorbic acid. That exact same molecule is sitting inside the package of hot dogs in your refrigerator, listed as a preservative. And that very same molecule was once used in darkrooms to develop black and white photographs, curing a disease that killed thousands of sailors, keeping meat from turning brown, bringing an image to life on film. Same molecule, same formula. But here is something that might surprise you. All three of those jobs rely on one single action at the atomic level. This is how a field of corn becomes white pharmaceutical crystals, and how that one action makes all of it possible. Ascorbic acid gives away electrons. That is the entire mechanism. It hands off an electron to whatever is nearby, and the outcome depends on who receives it. In your body, it donates electrons to unstable molecules called free radicals, neutralizes them. That is why your body can build collagen. Without those electrons, connective tissue falls apart. That is scurvy. In a hot dog, it donates electrons to reactive oxygen, stops the chain that turns meat brown and makes fat rancid. In a darkroom, it donates electrons to silver halide crystals on film, reduces silver ions into metallic silver. Where enough silver accumulates, the image appears. Same action, three outcomes. The only variable is the receiver. For centuries, nobody knew what was missing. Sailors on long voyages watched their gums bleed, their teeth loosen, their wounds refuse to close. The body could not make collagen without an electron donor, and nothing in their diet provided one. In 1747, a Scottish surgeon named James Lind gave scurvy-stricken sailors citrus fruit. They recovered within days. The British Royal Navy took 48 years to make lemon juice mandatory. In 1928, Hungarian biochemist Albert Szent-Györgyi isolated a substance from animal adrenal glands and later from paprika peppers. He called it hexuronic acid. Walter Haworth determined its structure and they renamed it ascorbic acid, Latin for without scurvy. Both received Nobel Prizes in 1937. Then you trace the supply chain and the oranges disappear. The molecule that cured scurvy starts as a field of corn. Not oranges, not lemons, corn. The corn is processed into starch. Then enzymes break the starch into glucose, pure simple sugar. That glucose is the sole raw material. The same glucose that competes with corn syrup, ethanol, and biofuel for the same crop. The original method was the Reichstein process, developed in 1933. Today, specialized bacteria do the heavy lifting through a two-step fermentation. First, a strain called Gluconobacter oxidans converts glucose into sorbitol and then into sorbose with a conversion rate near 100%. Second, a mixed culture of Ketogalenicidinium vulgare and a bacillus species converts sorbose into 2-keto-L-galonic acid. The fermentation tanks hold the mixture at around 30 degrees Celsius for 3 to 4 days, pH controlled between 6 and 7. Overall yield from glucose to the final intermediate runs at roughly 60%. At this point, what started as yellow corn exists as a pale chemical intermediate in a steel tank. A final chemical step, acid rearrangement, converts it into crude L-ascorbic acid. Then comes purification. The crude acid is dissolved, filtered, and crystallized. The crystals are washed and dried into a fine white powder. If you have ever tasted raw vitamin C powder, you know the sensation, a sharp, sour bite that dissolves on your tongue before you can swallow. The output is graded by purity. Pharmaceutical grade sits above 99.5%, supplement tablets and injectable solutions. Food grade runs above 99%, the preservative inside your hot dogs and orange juice. Feed grade goes into livestock nutrition. Technical grade goes into water treatment. Same molecule at every level. The only difference is how many impurities remain. And that film connection is not hypothetical. A formula called caffeinol instant coffee, vitamin C powder, and washing soda is still used by the analog photography community. 3 household ingredients. Total cost pennies. The ascorbic acid donates electrons to silver halide crystals, reducing them into metallic silver. Where light hit hardest, the most silver accumulates. The image appears. The molecule does not care whether it is inside a bloodstream or a developing tank. It donates electrons either way. Here is where the story flips. Almost every mammal on Earth manufactures its own vitamin C. Dogs, cats, cows, goats, mice. Their bodies produce it internally. Humans cannot. We carry a gene called GULO. that coded for the enzyme responsible for the final step of synthesis. Roughly 61 million years ago, it broke. If it still worked, no human would ever need a supplement. No sailor would have died of scurvy. Some bat species have managed to reactivate their broken GULO gene during evolution. Humans never did. Then there is the supply chain. More than 80% of all vitamin C produced on Earth is manufactured in China, 148,000 metric tons in 2024. One country controls the pipeline for a molecule that nearly every human depends on. Linus Pauling won 2 unshared Nobel Prizes, chemistry in 1954, peace in 1962. The only person in history to do that. Then he spent 25 years promoting vitamin C megadoses. In 1970, Vitamin C and the Common Cold became a bestseller, built on anecdotal evidence. He and surgeon Ewan Cameron claimed high-dose vitamin C extended survival in terminal cancer patients. The Mayo Clinic replicated the study 3 times. All 3 trials found no benefit. Side effects of megadosing included diarrhea, kidney stones, and iron overload. Pauling died of prostate cancer in 1994 at 93. He had been taking 18,000 milligrams of vitamin C daily, 300 times the recommended amount. The question Pauling asked was not settled. It was reframed. Research from the National Institutes of Health has shown that intravenous vitamin C produces blood concentrations 30 to 70 times higher than oral doses. At those concentrations, the molecule behaves differently. Phase 2 trials have tested high-dose intravenous ascorbic acid against cancers including glioblastoma and leukemia. Results are not conclusive, but the question shifted from whether vitamin C matters to how much and by what route. Pauling is gone. The molecule is still here. Laboratories are still asking his question, with a syringe instead of a pill. Next time you hold that tablet, you are holding an electron donor, a molecule giving away parts of itself, in your blood, in your food, on strips of film, since before you were born. 61 million years after the gene that could have made it inside your body went silent, the molecule has not changed. What changed is what you see when you look at it. That tablet in your cabinet, that ingredient on your hot dogs, that photo on your wall. One molecule, one action, giving electrons away wherever they are needed. That is the process. We reveal how things actually work one story at a time. If there is something you would like us to explore next, let us know. Until then, trust the process.